Lysosomes. Lysosomes are membrane-enclosed vesicles that form from the Golgi complex. Inside, as many as 60 kinds of powerful digestive and hydrolytic enzymes can break down a wide variety of molecules. Once lysosomes fuse with vesicles formed during endocytosis. Because lysosomal enzymes work best at an acidic pH, the lysosomal membranes include active transport pumps that import hydrogen ions. Thus, the lysosomal interior has a pH of 5, which is 100 times more acidic than the pH of the cytosol, than the systole. The lysosomal membrane also includes carriers that move the final products of digestion, such as glucose, fatty acids, and amino acids, into the systole. Lysosomal enzymes also help recycle worn-out cell structures. A lysosome can engulf another organelle, digest it, and return the digested components to the systole for reuse. In this way, old organelles are continually replaced. The process by which entire worn-out organelles are digested is called autophagy. In autophagy, the organelle to be digested is enclosed by a membrane derived from the endoplasmic reticulum to create a vesicle called an autophagosome. The vesicle then fuses with a lysosome. In this way, a human liver cell, for example, recycles about half of its cytoplasmic contents every week. Autophagy is also involved in cellular differentiation, control of growth, tissue remodeling, adaptation to adverse environments, and cell defense. Lysosomal enzymes may also destroy the entire cell that contains them, a process known as autolysis. Autolysis can occur in some pathological diseases and conditions, and is also a response for the tissue deterioration that occurs immediately after death. As we just discussed, most lysosomal enzymes act within a cell. However, some operate in extracellular digestion. One example occurs during fertilization. The head of a sperm cell releases lysosomal enzymes that aid its penetration of the oocyte by dissolving its protective coating in a process called acrosomal reaction. The clinical connection. Some disorders are caused by faulty or absent lysosomal enzyme. For instance, Tay-Sachs disease, which most often affects children of Ashkenazi, Eastern European Jewish descent, is an inherited condition characterized by the absence of a single lysosomal enzyme called Hex A. This enzyme normally breaks down a membrane glycolipid called ganglioside, or GM2, that is especially prevalent in nerve cells. Once again, this enzyme normally breaks down a membrane glycolipid called ganglioside, GM2, that is especially prevalent in nerve cells. As the excess ganglioside accumulates, the nerve cell functions less efficiently. Children with Tay-Sachs disease typically experience seizures and muscle rigidity. They gradually become blind, demented, and uncoordinated, and usually die before the age of 5. Tests can now reveal whether an adult is a carrier of the defective gene. Peroxisomes. Another group of organelles similar in structure to lysosomes but smaller are the peroxisomes. Peroxisomes are also called microbodies. Microbodies contain several oxidases, enzymes that can oxidize, remove hydrogen from atoms, and repeat. Peroxisomes also called microbodies, contain several oxidases, enzymes that can oxidize various organic substances. For instance, amino acids and fatty acids are oxidized in peroxisomes as part of normal metabolism. In addition, enzymes in peroxisomes oxidize toxic substances such as alcohol. Thus, peroxisomes are very abundant in the liver, where detoxification of alcohol and other damaging substances occurs. A byproduct of the oxidation reaction is hydrogen peroxide, a potentially toxic compound. However, peroxisomes also contain the enzyme catalase, which decomposes hydrogen peroxide because production and degeneration of hydrogen peroxide occurs within the same organelle. Peroxisomes protect the other part of the cell from the toxic adverse effects of hydrogen peroxide. Like mitochondria, lysosomes can self-replicate. New peroxisomes may form the pre-existing ones by enlarging and dividing. They may also form by a process in which components accumulate at a given site in the cell and then assemble in the peroxisome. Proteasomes.